In a natural world increasingly dominated by human impacts, including climate change, rapid urbanization, and deforestation, it is very valuable for ecologists to have metrics of the overall health of an ecosystem. In aquatic ecosystems, macroinvertebrates can serve as an accurate bioindicator of the overall ecosystem health. Studies have shown that downstream from human disturbances to aquatic environments, benthic macroinvertebrate populations are harmed both in terms of biodiversity and abundance. For our study, we sought to assess water quality and the ability of different aquatic locations with varying levels of human interference in Sandstone Ranch to support diverse and abundant macroinvertebrate populations. We examined both undisturbed sites in which human impact has been minimal to none, as well as locations that have been altered by humans, including a dam site and a gravel dumping site. We hypothesized that human disturbance of an aquatic ecosystem affects water quality and macroinvertebrate biodiversity, and we predicted that areas of aquatic ecosystems that have been disturbed would have differing depth levels that cause unnatural flow and poorer water quality, and therefore have less macroinvertebrate biodiversity than undisturbed areas. After data collection, we conducted several different types of statistical analysis to determine significance. First, we concluded that there was no significant difference in values for pH, conductivity, and dissolved oxygen, which signifies that the abiotic water quality indicators are consistent between the disturbed and undisturbed sites. We then analyzed data for the macroinvertebrate diversity and abundance. Pie charts of the macroinvertebrate species found in the disturbed and undisturbed areas can be seen on the poster. The major findings from these charts were that there were many more mayflies in undisturbed than in disturbed areas, and that there were many more horsehair worms in disturbed areas than undisturbed areas. This was interesting as mayflies are considered to be indicator species as they are very sensitive to environmental disturbances. We continued our macroinvertebrate analysis by conducting a test that accounts for both diversity and abundance of species. We found that the values for diversity and richness were larger in undisturbed sites than disturbed sites. However, statistical analysis showed that there was no significant difference between the disturbed or undisturbed sites. In conclusion, our hypothesis was somewhat but not fully supported, as there was no statistically significant difference between water quality or macroinvertebrate diversity and abundance between the disturbed and undisturbed sites. However, our data suggests that there might be a difference in macroinvertebrate diversity if more data was gathered. Therefore, we are encouraged by our results and hope to continue our examination of these trends in the future. By looking into both water quality testing and macroinvertebrate sampling, we hope to work towards having a better understanding of human impacts on the health of stream environments.